lovely to catch up with you, Jeff. First question to fire away for you is, so you have 30 years of clinical operations experience, including 13 years of experience in small interfering RNA space and other therapeutic modalities too. From your 4 years in the gene editing space so far, what do you consider to be the key challenges of running clinical trials with this modality?
I think that there's an importance or a responsibility of the permanence of this biology, and it gets a lot of the principal investigators and study teams excited. But there's still a tremendous learning curve that I think we're all on, be it regulators, ethics committees, patients, and families, to understand the concept of permanence and how do we get folks to understand kind of the rigor that goes in before we're even able to get to a clinical trial. I think you and I talked about this in the past. I would challenge gene editing companies to define themselves as educational companies that are also developing new medicines in this place, so that we really put that emphasis on transparency and education across all of those stakeholder groups. And they may even grow, in the groups. But I think it's important for folks to get a real true understanding before participation being, again, across those domains, institutions, individual departments, teams within departments, and of course, as I stated earlier, those considering treatment option of clinical trial participation. So those patients, the families, whatever that network is to support that decision making, really that's our responsibility in the education there.
So in those 4 years since your first involvement in this field, how have you seen it evolve in that respect, and what have been the key milestones along the way?
I think that there remains a hesitation. So Intellia wasn't the first. We're not going to be the last. I think the hesitation it is still tied to the fear of the unknown and some of the things that we just talked about. But I would say there's been a tremendous learning from the Regulatory and Ethics Committee side when it looks at the work and the rigor that goes around not just the identification of the target, but more importantly, the representation of lack of off-target editing, or where this goes throughout the body, but showing that it is safe beyond the target organ. And there's both in vitro, in vivo, animal work, and a lot of information work. And this is an emerging field that maybe I'm not really an expert in this area, but I would say, the more that we can share as an industry across these methodologies, and an understanding at that level of the national health authorities, the better. We've gone down in the number of questions. When I first got here, it often took 2-3 rounds of questioning from regulators, and 7 to 9 months to get an understanding to afford the trial to start. There remains some of that learning curve taking place. But I would say there is a commitment to this technology and a commitment to learning. So a lot of the regulators do try to stay within certainly any guidelines they have for review timeframes, but also pretty timely in getting back to us with questions. But
you mentioned a couple of times there the fact that this is an emerging field. I suppose in that we see lots of emerging technologies in research and development. But what do you think some of those key challenges are for people in the field to take those emerging technologies and actually translate them into viable therapies?
Well, I would say delivery to, we all are kind of building our companies on delivery to the liver, or that's a broad statement, but many of these are going to the liver. And that's a valid place to tackle a number of diseases. I think we're going to get to that new step very soon, extrahepatic. And again, that learning curve is going to start all over again. Okay. So how do we take the knowledge on educating those regulators and ethics committees and a priori make sure that we are, I think, more anticipating and or finding forums where we can educate within the regulatory space. I would also, I think this is an opportunity. I don't want to call it a challenge, but you also said, where are some expectations in the field or things that are exciting? I think there's a flip side to the education. It's not all bad. I would say when we look at the FDA, the EMA, MHRA, they're really starting to explore and hold conversations with companies like Intellia on platform approaches, much more platform approach. From a business side, we all understand the luxury of a platform and being able to kind of accelerate the work that takes place within an Intellia or other company. Now the regulators are taking that on to say, all right, how can we harness that as regulators and actually look at resources, efficiencies, and cross-referencing to a different degree than they've ever done before. And we are seeing that in some of our dialogue with the agencies that I've mentioned.
Thinking ahead to the 5th Genome Editing Therapeutics Summit later this year, for which you'll be delivering a keynote talk, sharing some insights from your work at Intellia around development and operational challenges associated with making the leap to phase 3 studies. As a sneak preview for this talk, I wonder if you could provide me with an insight into, again, where those key challenges lie with taking these leap and where those opportunities are.
I think with regard to internally at your company, again, this is not a page that's unique to gene editing, but there probably is some details there that is more unique to gene editing, is really making sure across the stakeholder group, the understanding of going from 4 centers or 5 centers in a phase 1 study to 100 centers in a pivotal trial, and really what is the importance of your commitment to really ensuring sponsor oversight, the appropriate CRO oversight, the appropriate vendor oversight in other areas. We want the agencies want these molecules to work. Our responsibility is then that you're building the infrastructure, the team knowledge, from a compliance perspective, a quality perspective, quality by design in particular, from the beginning, and partnering with those departments, compliance and quality. They're your friends. They're not your non-friends. I don't want to say enemies, but they're there to make sure that when you're submitting these documents at the end, or you're filing your NDAs, there will be inspections, and how do we make sure we're prepared for that? And that is something I certainly learned with my small interfering RNA experience, but I think now more so than ever, with some of the innovation, we have the responsibility to make sure that those things are in place, because the regulators are trying to take a look at these technologies, and they know they're coming. I think there's that piece. I think a second piece that I'm really excited about is how do we educate the ethics committees as well? What's unfortunate is when the work around COVID met its goals, institutions tended to fall back to their 30 or 40 years of sequencing and reviews. And there's no real incentive, I think, within an institution or that side of the responsibility or the chain of running trials yet to innovate in those spaces, or even just look at efficiencies. So that to me is an area that is bigger than Intellia, bigger than everyone at this summit. But how do we actually take a look at that? And there's a few things that are starting to emerge. There's a small group that Intellia and several of the members here at the summit are a part of. It's called the Precision Health Alliance, and it's centralized through Duke University. I think that we're in the charter formation stage, making sure the legal teams all understand what do you mean. These gene editing companies are coming together with Duke leadership to tackle some of these things. How do we get comfortable with that? That'll take a little bit of a while, and that's okay. But one of the initiatives that we've identified within the Precision Health Alliance is this education and kind of a little bit more at the institutional level. These molecules are coming. Hundreds more studies will come. Are the institutions set up just even tactically to approach this? And then once these molecules start to emerge into the clinical space, what will it mean for the business units there that you might be looking to evolve with this technology? Chronic administration of a product that then becomes a single dose administration will disrupt a business unit or a department within an institution. So we have to take a look at some of that as well. But I think, again, through commitment across these companies, I think that will help institutions take a look at some of these areas. Will gene editing be done in an outpatient clinic setting? I think that's interesting, Nick, for you to find the companies that their business model is closer to that as well, and start to gather some of that information. When you look at a lot of these portfolios are still defined maybe by rare diseases, where it's likely the editing infusion or whatever that delivery mechanism is going to take place in an academic research institution. But this will be interesting to watch when we get to some of the indications that are not genetically defined, and where will those therapies ultimately be administered. I think that's something for us to watch in the next decade as well.
And finally, it's still a few months away, but why are you excited to be involved in the summit this year, and what are you hoping to get out of your time with us?
Well, it'll be my first one, so I'm just kind of excited being a newbie, and I appreciate all the work that Handsome Wade has done to date to kind of put these types of touch points together for people. I know this isn't the only thing you guys do; you do many of these things, so thank you for that. I think really trying to find those spaces where we can share, even if we're in a competitive space. How can we do that for the betterment of the patients and families we really are all lining up to serve? I think there's going to be other areas, like I said, that I'm intrigued on. When do we start to see people get their second edit? Because they've gotten an edit for a rare disease, and then 6-7 years from now, they're getting an edit for a more common type of disease that runs in the person or in their family. Some of these emerging areas as to how that will work. I think another area I'd like to kind of hear about is when there are standards of care, and good standards of care, but maybe there's still room for improvement. This is a ethical space that we are finding ourselves in, and I'm sure others in this space will find themselves in, and how to kind of take a look at this approach in a different way. Controlled arm studies comparing two products, etc. It's been very successful. It's been the way that regulators and ethics committees have a familiarity with, and really kind of leads to data that helps in decision makings and approvals. But we find ourselves getting a lot of, well, why should a trial be run? There's already an accepted therapy here. Why should someone move to now an unknown, and taking a look at that? So I think this is an appropriate set of questions that we're all going to be facing. And how do we afford true empowerment of that patient and family once we've hit that education bar that we talked about earlier in this conversation, so that a patient can make that decision to say, I am on a product that's approved, but I am willing to actually take this next step for me, or for the betterment of my family members, or general knowledge. I think this is an area for us to work together on.
Lovely. Thank you very much for your time and insights today, Jeff. I look forward to catching up properly in December.